Okay, hello, grade 11s. Continuing on with our bacteria notes. So, last time we talked about archaea, so let's talk about bacteria. And we talked about the differences between archaea and bacteria. Remember, the cell membrane is the key difference, they have a different cell membrane. We talked about some of the characteristics of bacteria, that they have no membrane bound organelles, they're very small. They have flagella. Some of them have flagella to move. Some of them have these pili, these little hairs, so they can attach themselves to each other or to other things. And they come in there's three general shapes to bacteria cells. So the shape is called coccus, and these are spheres. They look like little spheres. Where are some? Here we go. Let's see if we can figure out. There's some coccus. See, they're little spheres. They're stuck together, little spheres. There is bacillus, and these are rod-like. So there we go. That's bacillus, rod-like. So that's just the shape of the cell. And there is spirillus. Spirillus, they look like spirals. There they are under a microscope. They look like spirals. And depending on the species and whatnot, they will have these different shapes. So there's some examples here. I don't know why that sphere is in the way, but there you go, whatever. So Streptococcus, look at that. In the name is the shape. These guys don't necessarily have, that's not for all of them that we do that, but Streptococcus causes strep throat, so it's a little coccus cell. I wonder if you guys can look back on that microscope lab that we did. What did the bacteria look like under the microscope? Which shape were they? It's hard to tell because they're very small. They were yogurt bacteria, so I don't know which ones those are, which ones make yogurt. I think they were either bacillus or coccus. Let's talk about also, so they vary in the shape, so the cells vary in the shape. You got spirillus, bacillus, and coccus, and they also vary in their cell wall. So not only archaea have different cell walls, they have the lipids are different. Sorry, that's not the cell wall, that's the lipid, the membrane, but the cell wall. So there's the membrane, and then there's the cell wall. Bacteria have a cell wall, which is made out of protein, and it's made out of sugars. Peptidoglycan, actually, is what it is. And we tell the walls give them sort of something different function, different abilities, and so when we're looking at them, studying them under a microscope, we want to know what kind of wall they have, and we've dubbed these walls gram-positive and gram-negative. And that name refers to when you're looking at them under a microscope, you use a stain so that you can see the cells better. If you didn't use a stain, you wouldn't be able to see them because they just would be clear, they'd be transparent. You know how we looked at those cells and they were pink in the microscope? Well, they were stained. They were stained pink. And they were actually stained with this gram staining. So there's a little bit about that. So gram staining. So basically how this gram staining works, do we have two pictures of the wall? So here we go. There's the two different types of wall. And basically gram-negative walls, the layer of peptidoglycan, which is that sugar, is a lot thinner. So there we go. Those are gram-negative. And they have an extra layer, an outer membrane. So they have an inner membrane and an outer membrane. That's the lipid bilayer membrane that all cells have. And they've got, like transport proteins in them. And so these guys have an extra membrane. Those are gram-negative cells. Gram-positive cells have way more peptidoglycan. They got like a lot more armor, a lot more cell wall, and they don't have that extra outer membrane. And how we tell them apart is with this gram staining. And so what happens in the gram staining, we use a gram stain, and it actually contains two different dyes in it, a crystal violet dye, which looks purple, and a saffron dye, which looks red. Now, the gram-positive bacteria what will happen is the crystal violet gets stuck in these layers of peptidoglycan. And as a result, they look purple when you look at them under a microscope. Gram-negative, the crystal violet doesn't get stuck because there's a cell membrane here, but instead the safrarin red, is that what it's called? Yeah, safrarin dye, the safrarin red gets stuck in the lipids. That's what it's designed to do. And so the lipid layer absorbs the red safrarin, and it appears red or pink under a microscope. Gram-negative, so they look pink. So the bacteria that we looked at in our microscope lab, if you remember, were pink. So those were gram-negative bacteria. So yogurt bacteria are gram-negative. That's your short video for today. We're winding down the bacteria thing. By today, we should actually be finished. We're going a little too slow. We're running out of time. So hopefully we'll be able to catch this up when we come back. I don't want to make this video too long, so we're not going to go into reproduction because that's a whole big thing, and we'll do that tomorrow. And we're also going to start prokaryotes. So notes for prokaryotes will start to appear soon. Goodbye.